Thank you, Brian. It's a pleasure to be here. I'm Anand, and I make biosensors for neural drugs. Most of you take some kind of a drug that affects your brain and you know the desirable short-term effects. But what we don't know as well is how the long-term effects play out. For example, why is nicotine neuroprotective? Why does schizophrenics self-medicate by smoking? For depression, why do people suffer while drugs take often weeks to work? For pain, we've used opioids for millennia, but we can't seem to make less addictive versions. My research starts at the common beginning. How does the drug get around your body? How much of it gets to the brain? And how long does a dose stick around? The journey of a drug starts right after you take it. It gets absorbed into your blood, gets broken down in your liver. Some of it makes its way to the brain. From there, some activates receptors and neurons. Even within cells, there are compartments where the drugs can accumulate, leading to stronger, prolonged effects. The problem is that this distribution varies over space and time at each level, and it looks different for different people. We need a way to measure the drug itself at each of these levels to learn how it works. But we can't see the drug at the molecular level. Seeing is believing, so I evolved biosensors for opioids like fentanyl. These biosensors take two proteins from nature. The magenta one is from bacteria, and it clamps molecules like a Venus flytrap. The other is green fluorescent protein from jellyfish, which fluoresces under the right conditions. We join these two proteins so that the drug binding triggers an increase in the light from the green protein. This allows us to see the drug down at the molecular level, and we can even see it at a single mouse neuron, as you see in the top right. Now we can tweak the building blocks of the protein to selectively bind the drug. I converted the one in the middle for nicotine to detect opioids like fentanyl. Because fentanyl is so potent, we needed to be sensitive to a few nanomolar, and that's like dissolving this lethal 2 milligram dose in about 10 bathtubs worth of water, pulling out a few drops, and still fishing out a signal. Now, don't actually do that with fentanyl, but you get the idea. Now, this whole scientific journey is uniquely Caltech. We make the proteins from scratch here, put the DNA in viruses, and use those viruses to deliver the sensor to neurons in living animals. I work with chemists, biologists, medical engineers across campus to make this all happen here under one roof. We can then share the DNA for this sensor with outside labs, and they can plug and play in their own model systems. Now I want to solve the problem of drug monitoring for people using these very biosensors. The aspirational goal is to make wearable devices like a Fitbit or Apple Watch, but for drug monitoring so that you can tune up and down your dosage. In a budding collaboration with Wei Gao's lab, we want to functionalize a sweat patch, and the hope is that personalizing doses will prevent risk of addiction while maintaining a high quality of life. Thanks.